Okay, we left off this. I did a little different with this class. I told you guys under what conditions the trip operon will be on and off, and the lactose operon will be on and off. So what we're going to do first is I want to recap the tryptophan operon, and I want to reiterate and reemphasize a few things to be sure that everybody's clear on what this is doing, who is doing this, and what it's used for. Tryptophan is one of the 20 amino acids. You get it from things like turkey and chicken and other types of poultry. That's why I told you often hear about tryptophan a lot during Thanksgiving. Maybe not a lot, but maybe you just name drop once or twice. So tryptophan is one of the 20 amino acids, and it's actually an essential amino acid. Our bodies cannot make it. So the only way that we can get it is if we eat it. But we need help digesting food. We can't do it alone. And so we actually have bacteria that live within our own bodies that help us digest food and get all the nutrients from it. They break down carbohydrates, they break down proteins, they break down fats. Well, because tryptophan is one of the 9 essential amino acids that we do not make, we need to get in our food. The bacteria that actually reside in your intestines help you obtain that tryptophan and use it for other purposes throughout your body. So I want to make sure you all are clear that what we're discussing is taking place in bacteria. It is not taking place in you. It's taking place in the organisms that live in you. And so the tryptophan operon is normally turned on. We eat a lot of food that has tryptophan in it. One of the main foods is birds, chicken, turkey, maybe duck now and then. You go to Bar Taco, you get your little duck taco, stuff like that. So normally the tryptophan operon is turned on. What it is going to produce are enzymes that help to get the tryptophan from the food that you eat. Whenever tryptophan is in good supply, you will turn it off. So what I would like to do is I want to draw out, as I have done on the whiteboard, but step by step, what each of these operons looks like. And there's only 2. I want to draw it in the on position, the off position for tryptophan, and then the off position, the on position for lactose. So let's get started in drawing what the tryptophan operon appears to, at least how it looks when it is in the on position, which for tryptophan is the normal position. So I'm going to keep the colors consistent with yesterday. You see these zigzags I'm drawing? This is the DNA double helix. So these are the regulator genes. For the sake of time and space, I'm only going to label one of these operons as I go forward throughout today. I'm not going to label the silver part regulator and the red part promoter, blue operator, green structural. This next set of DNA is just junk, okay? It's nothing. It's just showing you that the regulator is not part of the operon. If you look at your drawing from yesterday, you'll see that according to the definition, the regulator is not actually part of the operon. So this red part really has no purpose except to space it apart. Then we have this red portion, which is going to be the promoter. This is where RNA polymerase will bind. I'm going to ask you guys about RNA polymerase in a moment. We went over it yesterday, see if you recall. Okay, here's the operator. This is basically the switch, the on-off switch for the operon. I should probably label these. Promoter, operator, and these are the structural genes. This is where the actual instructions are to make enzymes. In this case, these are the enzymes that are able to get tryptophan. Get the tryptophan from the food that we eat. Get it out of there. You have to be able to metabolize it. So the question was, what do I mean by get the tryptophan? Get the tryptophan by getting it from the food that we eat. Chicken nuggets, Chick-fil-A sandwich, Thanksgiving meal, any of the various foods that have tryptophan in it. Okay, now, one thing I'm missing here is a repressor. And before I get to that, I want you guys to look at the operator. You see that little square-shaped notch in there? I drew that on purpose. That is where the repressor plugs in. The operator is basically the on-off switch. Well, a repressor, as we went over yesterday, the definition is it subdues, or it stops, or it halts. Take a look at how I draw my repressor. Can that little knob on that repressor fit in that operator the way it is right now? Can a circular peg fit in a square hole? No. So if this repressor is not being used, do you remember what we call it? It's inactive. This is known as an inactive repressor. So Reagan, if the repressor is inactive, does that mean the operon is going to be on or off? Off on. Why would it be on? The repressor can't touch it. So Dan is a mean green fighting machine when I don't do this. I am the repressor. When I do this, I'm holding it down. Right now, am I active or inactive? I'm active. I'm actively stopping him from fighting people. When I let go, is he free to fight people? Yes. So is he on or off? He's on. If he's on, I'm the repressor. What am I? Inactive. If I am active, what is he? He's off. The operon is either on or off. The repressor is either active or inactive. So let's try this again. I'm active. What is he? Off. I'm inactive. What is he? That's how it goes. So, Reagan, the repressor is inactive. What does it say about the operon? So don't draw this. Just watch. This, can that fit in there right now? No, can't happen. So the repressor is inactive. What that means is the enzyme that we call RNA polymerase, and this is where I want to ask you, can anyone tell me, what does RNA polymerase do, or what does it make? Ryan? From what? Very good. RNA polymerase will make RNA from DNA. So here's the next big question. Risa, is there anything that is getting in the RNA polymerase's way of scanning the structural genes and making RNA? Do you see any repressors that are in its way? No. So watch this, guys. You can't do this on your drawing, so just watch. RNA polymerase is going to make it past the operator, and now it's going to scan the structural genes. And what's it going to produce, Ryan? That's right. So it's going to produce an RNA, mRNA. And what do you get? That's transcription. What are you able to produce from an mRNA template? Go ahead. No, you use the mRNA, the tRNA, the rRNA. They all work together to make what? Proteins. And what's that process called? Translation. Now, to you, someone who might not be a total whiz when it comes to biology, you're like, why is he drawing a bunch of pink squiggles? These squiggles are actually proteins. Remember, proteins have to be in a twisty contorted, three-dimensional shape to actually work properly. And what's so special about these proteins? These proteins are enzymes. That get tryptophan. And this is how the trip operon operates when it is on. Duncan, real fast, what type of organism is doing this? A what? No. Prokaryotic. The name of this lesson is prokaryotic gene regulation. Prokaryotes. Bacteria. Casey, real quick, where is this living? In your intestines. You guys have trillions, with a T, trillions of bacteria living in your intestines. It's a lot. And to kind of get a little nasty, it's what makes your farts and your poops smell. It's the bacteria. It's the byproduct of what they do. Okay. So, are there any questions of, I didn't write this down, so that's my fault. Let's get that fixed. This is the tryptophan operon. So when it's on, trip operon on. Here's the basic logic, guys. And it really can be boiled down to this. Hartley, I want you to finish my sentence. There is no tryptophan, so what are you going to do? Yeah. It's really that simple. There is no tryptophan, so you're going to synthesize it. But keep this in mind, everyone. There are 9 essential amino acids. Essential means our bodies don't make it. We have to eat it. These bacteria help get it from the food that you eat to be used by your own body to build muscles, to build hemoglobin, to build keratin, all that stuff you get from Chick-fil-A, chicken nuggets, chicken breasts, Thanksgiving turkey, all that good stuff. Yeah.
If we didn't have all of the trillions of bacteria in our intestines, like what would happen?
We're dead. There's a notion, you know what a woolly mammoth is? A woolly mammoth is a prehistoric relative of the elephant. It went extinct about 10,000 years ago, give or take a few thousand years. It's gone. Well, its closest living relative is an elephant. Well, thanks to some recent finds, scientists have found perfectly preserved woolly mammoths, like babies, in the permafrost in places like Siberia, Scandinavia. They're frozen in time. It's like Captain America style. They're frozen in time. They still have their skin. They still have their hair. They still have their toenails. They have everything. They even have tissue that's perfectly preserved. And we're thinking, okay, we could somehow regenerate this DNA of a woolly mammoth and make one. It's kind of like Jurassic Park, but with ancient mammals, not dinosaurs. The problem is, let's say that we do bring back a woolly mammoth. How is it going to survive without the gut bacteria? We call that your internal biome. A biome is a living environment, like numerous ecosystems together, like the rainforest is a biome. The desert is a biome. You have an internal biome. How are we going to keep these woolly mammoths alive if we don't have the bacterial, the intestinal bacteria to keep them alive? What do you think? They're probably dead. No. The elephants or the woolly mammoths are dead. The bacteria is dead. What is the closest living relative we could use that may have suitable bacteria to put in their intestines? Ayla? Elephants. Yeah. Now, something else that's kind of nasty, guys, is there are people that have conditions where they have hardly any bacteria in their intestines. So there's a procedure, and this is for real, where you take donor poop. You then separate the poop from the bacteria that's in the poop. Every time you poop, there's bacteria in there. That's kind of one of the reasons why it stinks. And what you do is you kind of boil it to get all the solid chunks liquefied. And then you put it up, you put some tubes up someone's nose, and you send the liquefied poop with the bacteria up their nose, and it goes down into their stomach, and it basically plants new bacteria into their intestines. It's like it's called a fecal transplant, and that's real. It gives the patient new healthy bacteria that they didn't have.
They knock them out though, right?
I hope so.
Put it up there.
Yeah.
The liquid poop.
Yeah, I hope so. Yeah. What's that?
South Park did an episode on
You, what episode? South Park. Oh, South Park likes to spoof things that are real. You ever see that South Park episode where? Hold on a second. I don't know what this means. Okay, so back to this. Let's talk about the trip operon when it is turned off. Oop, why am I writing it on there? Off. Okay, under what conditions will the trip operon be turned off? Trip is present. There's no need for more. All right, so let's redraw everything, guys. This will take just a moment. Oop, wrong color. Remember, what I'm drawing is the DNA of the bacteria. Okay. So there's the whole sequence again. We're going to draw it in the off position now. If you take a look at the whiteboard, you can kind of jump to the ending here. Here is our repressor. Currently, it is inactive. However, we have something new coming into the fray. This is called tryptophan, the amino acid, tryptophan. Now, do you kind of notice something about the tryptophan and the repressor? They're going to go hand in hand. Not only is tryptophan the amino acid that we're trying to make, but tryptophan is also going to serve as something called a co-repressor. If Dan is planning on robbing the bank and Jet is planning on being his getaway car, Jet is going to be considered a co-conspirator. He's just as guilty. Co means it's helping, it's part of it. A co-repressor is going to help the repressor. So watch this. Whoop, I did not mean to do that. Here we go. Okay, here we go, here we go, here we go, here we go. Locked in. Now, this repressor will no longer be inactive. Check out the little nodule on the top. It's in a square shape now. It is now active. And what it can do is it can plug into that operator and turn off the operon. The repressor is now active. So if I take RNA polymerase again, can RNA polymerase get past that repressor? Check it out. It's like a roadblock. It's not going to be able to get by. So this operon is turned off until all the tryptophan has been absorbed. So this operon is now in the off position. The tryptophan turned the repressor from being inactive to active. So I'm going to write that down. Tryptophan served as a co-repressor, switching the repressor from inactive to active. You know you have tryptophan when tryptophan actually turns on the repressor. Okay, I want to ask you guys now, before we move on to the second operon, what type of feedback loop is this where something is happening and you want it to stop? Annie? Negative feedback. If there is no tryptophan, you need to make it. If you do have tryptophan, stop making it. That's all negative feedback. So everything I have for the trip operon is over there on the left side of the whiteboard for you. Any questions, guys? Duncan, you hanging in there? How many days to the AP exam, Duncan? 109. Okay, if you guys are at least have this stuff down, and by the way, it is totally normal and natural to have to study this a couple times to really have it sink in and understand how it's working. So expect that. You should do that. Now, the second operon we need to learn about is called the lac Operon. Lac is short for lactose. I'm going to ask you some questions about lactose, and this is from the first unit back in September. What type of macromolecule is lactose? Carbohydrate, lipid, protein, or nucleic acid? Jasmine? It is a carbohydrate. Very good. You can tell because it ends with OSE, sugar. Now, carbohydrates come in different sizes. Is the lactose a monosaccharide, disaccharide, or polysaccharide? Ryan? Good job. It is a disaccharide. Okay, last question from our little blast of the past. A disaccharide is made up of two monosaccharides. My last question for now is what two monosaccharides will bond together to form lactose? Do you recall that? What plus what equals lactose? Jasmine? You said glucose? Not glucose? Okay. Anyone? Think back to September? It was like a lifetime ago. Sage? Which one? Yeah, galactose. That is one. I'll put GAL for galactose. What's the other one? No? No, not fructose. That is a monosaccharide, but not fructose. Jasmine? No, sucrose is another disaccharide. That's glucose plus fructose. Right?
I'm not sure, but no one said amylose.
Amylose is a polysaccharide. That's starch. Riley? Glucose. Jasmine, you said glucose and you said not glucose. Should have got the first answer. It was glucose. You said glucose, right? Yeah. It's glucose and galactose. Okay. Now, here's a question from Unit 3. What do we need for cell respiration? Like, what is the necessary reactant in order to make all that ATP? What do you have to have?
Oxygen.
Oxygen is a catalyst that it serves as a final electron acceptor for the breakdown of what? That's it, glucose. So here's what I'm going to tell you all. The whole purpose of the lac Operon is to obtain glucose. That is something very important. The reason lactose must be metabolized is to obtain glucose. That is the purpose. You're not just breaking down lactose just because it's there. You're breaking down lactose to get glucose. Now, this process, this operon, as Duncan said earlier, is in prokaryotes. It's in the bacteria in your intestines. Now, it is very, very weird that we, as adult mammals, you guys are pretty much adults now, adult mammals drink milk. Not only is it weird we drink milk from other mammals. Adult mammals don't drink milk, babies do. But over the centuries, our ancestors, depending on where your ancestors were on Earth, they would drink the milk of cattle. And eventually, some mutations developed within the E. coli in our guts that allowed them to process lactose. Not everyone can do this. Those people are called lactose intolerant. Like a lot of people that descend from India, they don't drink milk because cows are considered sacred, so you don't drink the milk of it. So most Indians are lactose intolerant. They don't have that mutation. Most of you do. So the whole purpose of breaking down lactose is to get to the glucose. So I'm going to ask you guys a few questions here, or I'm going to give you one question, and I'm going to give you the 4 options. And I have seen this question on the AP Biology exam before. So the question is, under what conditions will the lac Operon be turned on? I've seen this question on several years worth of AP exams. Here it is. Option A, high glucose, low lactose. B, high glucose, high lactose. C, low glucose, high lactose. D, low glucose, low lactose. What is the purpose of breaking down lactose? What are you trying to get, Sam? You're trying to get the glucose. Take a look at option A. This is what we're going to do in the month of March and April. We're going to be doing those practice tests, and we're going to be going over every single option. We're not only going to identify why the correct answer is correct, but why the wrong answer is wrong. Glucose is high. Lactose is low. Do you break down lactose when it's not there? No, it's not there. Hartley's deathly afraid of cockroaches, but she's not sitting there pounding her feet on the ground. Why? There's no roaches here. You don't address, you don't bother, you don't waste your time with what's not there. The glucose is high. The lactose is low. Do you need the lac Operon to break down lactose? No. So option A is not the correct answer. Here, part B, glucose is high, lactose is high. Now, there is lactose. But remember what Sam just said. The purpose of breaking down lactose is to get to the glucose. You already have glucose. Do you need to bother breaking down lactose? No, you don't. So B would also be a situation where you do not turn it on. But Mr. Thorson, the lactose is high. Yeah, I hear you. But keep in mind as to why you're breaking down lactose. You break down lactose, get to glucose. Glucose is already there. C, the glucose levels are low, the lactose levels are high. Would you turn it on then? Yes. Correct answer is C. There is no glucose, there's a lot of lactose. So break down the lactose, you'll get to the glucose. Okay, so let's draw this out. This is going to be, normally, The lac Operon is in the off position. This is normal. Here we go. Regulator. Remember, guys, all these squigglies are DNA. Void filler DNA. Promoter. Operator. Structural. Okay, I want you to check out that repressor right now. Is the repressor hooked up to the operator? Yes or no? So is the repressor active or inactive? Is the operon on or off? Good. Can RNA polymerase get past the operator? If I draw this RNA polymerase, as you learned yesterday, it goes onto the promoter. Can it get past the operator? No. It's like running into a wall. So the repressor is turning the operon off. In the absence of lactose, the lac Operon is off. Yes, Annie.
So, this is a little bit related to something earlier, but if, like, when you're a kid and you have milk and you can process lactose, what changes when you're an adult? That's like not a thing you do.
Your body changes just as you grow, and its needs for all that calcium and other nutrients wanes. And so the bacteria that you have is looking towards metabolizing other nutrients to help you grow. Yeah.
Okay, so like, let's say you drink like chocolate milk or something, and there's glucose from the chocolate or like the sugar along with the lactose from the milk. Is your body gonna process the glucose first?
The glucose will automatically get absorbed into your bloodstream because it's small enough, but the lactose is a disaccharide, so it needs to be broken apart before.
So it's just gonna stay in your stomach and your body.
Now, a monosaccharide is small enough to be absorbed into your bloodstream. Your intestinal walls are so thin that monomers can be absorbed. Lactose is a disaccharide. It's not a monomer yet. So your digestive system's job and the bacteria that's in your digestive system's job is to break apart things like a disaccharide or a polymer to their monomer form so that they can be absorbed. So while the glucose gets absorbed, the lactose needs to be digested first, and then it will follow the absorption.
So even if you have like enough glucose in your body, it's still gonna.
In that case, sure. Okay, the last one. We have about 5 minutes left, so let's get this last one in here. Now it's going to be in the on position. Okay, check out your repressor. It's inactive. What made it inactive? Lactose. Lactose fit into those binding sites and turned the repressive into an inactive form. So this person in which this bacteria lives just probably had a glass of milk or ate some ice cream. And now there's lactose in this person's digestive system. That lactose is going to adhere to the repressor and make it inactive, which is going to allow the operon to be turned on. Now that the operon is turned on, can RNA polymerase transcribe? Yes. So check it out. Watch. This brown blob is RNA polymerase. It's going to jump onto the promoter. It's going to scan the structural genes like so. And from that, you're going to be able to make mRNA. That's transcription. And then from the mRNA, you are going to be able to make proteins. Or actually enzymes, to be exact. These are lactase enzymes. Now, you all learned a long time ago that enzymes are named after what they break down. What do you think the enzyme lactase breaks down? Lactose. So that lactase will break down the lactose and get the glucose and use the glucose for cell respiration. Once all the lactose has been broken down, the repressor will plug back into the operator and turn the operon off because all the lactose is gone. Yes, Andrew? They'll denature. Once their purpose has been completed, they're going to just denature and break down. I'm sorry? What is what's purpose? What is lactase named after? Listen, enzymes are named after what they break down. What do you think lactase is named after? It breaks down lactose. If you are lactose intolerant, you likely lack the enzyme lactase. You don't have it.